Hello everyone. Today we are going to talk about behavioral genetics, and I'll be introducing the first half of the PowerPoint here. And I want to point out that we have covered some of the points that are made in this chapter in earlier PowerPoint presentations. So I'll be sending you back toward those earlier PowerPoints occasionally. So the first thing I wanted to show you, the first thing I have for you is a link to a video. It's here, but it's also in the web links in the list of your files for this chapter. And this is just a videotape of a border collie at 9 weeks old doing what border collies do. And the point is that even with very little time awake and aware on this earth, this border collie is performing all kinds of behaviors. And those behaviors are innate. These are encoded in their DNA, so they don't have to be learned. And quite a lot of our behavior and behavior of other animal species is just that. It's innate. In fact, there are three main sources of behavioral variations. There's genetics, there's environment, which includes all of the learned behavior, and then there's the gene and environment interaction, because we know that genes get turned on or activated by environmental variables quite frequently. So one of the questions that comes up a lot is, well, how much of that behavior is due to genes? And that's a problematic question to answer, especially if we're talking about individuals. What we have been able to do is begin to measure genetics effects on variability within a population. So for example, how much the variability of height in humans is associated with the genetics for height. And the answer to that is quite a lot. It's about 80%. But most behavioral questions are actually quite a lot less associated with genes. Usually about 50 to 60% variation is very high for behaviors. We still talk about it in the same way. We talk about heritability, and heritability is the proportion of the phenotypic variation in a population that is due to genetic variation. Remember, we're distinguishing now between phenotype and genotype. So what is visible to us on the surface, on the outside, is phenotype, and genotype is what's actually encoded in our genes. And heritability is h squared is equal to the variance in the genotype that's divided by the variance in the population. So that means that obviously the variance in the population is basically variance in the genotype plus variance associated with environment. And now we're trying to strip away the environmental issue and just look at the variability in the genotype itself. So this is a number that can be actually calculated. Problematically, though, the factors that are associated with the variability in the genotype and in the phenotype really are multiple. So you can have additive effects, which basically means that because you have genes, separate alleles, you have multiple of those. Sometimes if you have the gene for a very tall person, and then they also on their other chromosome have a gene for a very tall person, that makes them an extremely tall person, right? That's the additive effect of those genes. Dominance effects means that sometimes, for example, blue eyes and brown eyes are dominant over blue eyes. So even if you have a brown gene and a blue gene, you will end up with brown eyes. And epistasis. Epistasis is the interaction between alleles at different loci. So we know, for example, that there are multiple genes for eye color. One of the reasons that we get hazel eyes and they have such a large variation in them is through epistasis. So blues and brown coded areas in different genetic alleles result in different colors. A lot of the times, only the additive effects are reported because the genetic variation is too high to estimate D and I, right? So what that means is that heritability in the broad sense takes into account all of these effects, additive, dominance, and epistasis, but heritability in the narrow sense just looks at additive effects across the alleles. So what kind of behaviors do we see that are associated with genes? Well, mostly we're going to see relatively unvarying types of behaviors. Reflexes are a standard type, and fixed action patterns. We talked about fixed action patterns in the last chapter, so I'm not going to go over them again here. But let's talk about reflexes. Reflexes are a really basic form of elicited behavior. And elicited behavior simply means that there's a stimulus and then the behavior automatically happens. Okay, so the overall definition is a relatively simple automatic response to a stimulus. Reflexes are closely tied to survival. For example, startle reflexes when you're afraid, orienting responses in babies. So if you brush the cheek of a baby, they will turn toward your hand, and that is so that they will turn their cheeks toward the source of milk. And you can see by the picture in the middle, newborns actually have a grasping response, which is strong enough that they can actually hold themselves up by one hand. This goes away very, very quickly. But that grasping response is very important not to humans, but to our evolutionary ancestors, the primates, who immediately begin holding on to their mothers by grabbing onto her fur. One of the interesting points about a reflex is that it is activated and sometimes responded to without the brain actually being involved. And that's what's called a reflex arc. And that is a neural structure basically that leads from the sensory neuron, in this case, in this example, in the finger, leads directly up to the spinal column, so the back, and that is immediately turned back around to the muscles in an afferent neuron to move the finger out of the way. Only after they send the pulse back to the hand to move the hand is that is that pulse transmitted to the brain. It allows us to react immediately and automatically to dangerous stimuli in the environment. We don't have to think about these things. We don't have to consciously decide to pull our hand back. The reflex arc lets us do that without thought. That is clearly going to be a non-learned response or a biological response. And those are, of course, generally tied to survival. Fixed action patterns we talked about, similar to but more complex than reflexes. That's a fixed sequence of responses elicited by a specific stimulus. We talked about sign stimuli, which are releasers. Releasers are social versions of sign stimuli. That's the stimulus that sets the fixed action pattern into motion. So when we talked about the gray-legged goose and they're moving their eggs back into the nest, all they have to see is a roundish type object outside of the nest to trigger that fixed action response. Other examples, sign stimuli. Beta fish will fight beta fish automatically. That's an aggressive behavior. Male sticklebacks will also attack other male sticklebacks, but what they've found is that it's the belly, the red part of the belly that actually is the releaser in this case. If you actually bring a male stickleback, another male, into the territory, but you cover up the red part of their belly, that first male who is already there won't attack. Finally, major and minor genes are different, those that contribute differentially to the phenotypic variation. Major genes contribute to a majority of the phenotypic variation, and minor genes are those that are somewhat smaller. Remember that genes are portions of DNA that express traits, that create proteins or change behaviors, activities, etc. Major genes don't mean that they're necessarily longer, but rather that they have a greater effect on the phenotype. So that's all I'm going to talk about in this one. There's plenty of extra information in your chapter, as well as in the rest of this PowerPoint presentation, but this mainly deals with methods of studying genetic variation and examples of how we piece apart genes and environmental effects on phenotype. So feel free to go through this in detail, and also feel free to email me with questions that you have. See you next time.